Hi, we're the Q group, and I'm Mahela.
I'm Patrick.
I'm Tamai.
And I'm Emily.
What is addiction? Addiction is the inability of a person's body to properly function without a specific drug after becoming dependent on said drug. Addiction is comprised of two mechanisms, drug tolerance and withdrawal. For now, I will focus on drug tolerance. As the person continually uses the drug, larger and larger doses are required to feel the same initial effects of the drug. This is called drug tolerance. There are two different mechanisms involved in building up a tolerance to a drug. Pharmacokinetic tolerance, which is known as dispositional tolerance, and pharmacodynamic tolerance, which is known as reduced responsiveness. Pharmacokinetic tolerance is the ability of the body to upregulate the production of certain enzymes required for the degradation of the drug. An example of this would be the CYP enzyme, specifically CYP450. CYP450 enzymes are essential for detoxification of foreign chemicals and the metabolism of drugs. If a drug is introduced into the body and induces CYP450 enzymes, less of that drug will reach the reaction site. CYP450 enzymes most commonly affect ethanol, barbiturates, opiates, and benzodiazepines. The second mechanism of tolerance is pharmacodynamic tolerance, which usually results in changes in the number of receptor sites on a neuron. This is called downregulation. Downregulation is cellular decrease in the number of receptors to a molecule, such as a hormone or a neurotransmitter, which reduces the cell sensitivity to the molecule. This is the mechanism that makes the user require the drug in higher quantities in order to obtain the desired effects. Long story short, the neurons are desensitized to the presence of the drug.
Neurotransmitters are small molecules such as amino acids and amines that transmit signals from a neuron to a target cell across a synapse. Neurotransmitters stimulate neurons to fire while others inhibit firing. Neurotransmitters bind with receptor proteins on the membranes of the neurons with a specific lock and key mechanism. This just means that neurons in different pathways correspond to different types of receptors in their own family. So dopamine binds to dopamine receptors. Neurotransmitters are released in a process called exocytosis. When the action potential reaches the end of an axon, the depolarization causes calcium channels to open, which causes synaptic vesicles that carry the neurotransmitter to fuse with the cell membrane. This fusion allows the neurotransmitter to be released into the synapse. Now I will talk about neurotransmitter receptors. Neurotransmitter receptors are membrane receptor proteins that are activated by a neurotransmitter. A membrane protein interacts with the phospholipid bilayer that encloses the cell, and a membrane receptor protein interacts with a chemical in the cell's external environment which binds to the cell. Membrane receptor proteins allow cells to communicate with one another through chemical signals. In postsynaptic cells, neurotransmitter receptors receive signals that trigger an electrical signal by regulating the activity of ion channels. The binding of neurotransmitters to specific receptors can change the membrane potential of a neuron. This can result in a signal that runs along the axon and can be passed along a neural network. On presynaptic cells, the binding of a neurotransmitter to a specific receptor provides feedback and mediates excessive neurotransmitter release. Now I will talk about neurons themselves. A neuron is an electrically excitable cell that processes and transmits information through electrochemical signals using neurotransmitters and receptors. These signals between neurons occur via synapses, a specialized connection with other cells. Neurons can connect to each other to form neural networks. A typical neuron possesses a cell body that includes dendrites and an axon. Dendrites bring information to the cell body, have a rough surface, have no myelin, and branch near the cell body, while axons take information away from the cell body, have a smooth surface, have myelin, and branch far from the cell body. Now I will talk about the neurotransmitter process. In the first step, neurotransmitter molecules are synthesized from precursors under the influence of enzymes. In the second step, neurotransmitter molecules are stored in vesicles. In the third step, neurotransmitter molecules that leak from their vesicles are destroyed by enzymes. In the fourth step, action potentials cause vesicles to fuse with the presynaptic membrane and release their neurotransmitter molecules into the synapse. In the fifth step, released neurotransmitter molecules bind with autoreceptors and inhibit subsequent neurotransmitter release. In the sixth step, released neurotransmitter molecules bind to postsynaptic receptors. In the seventh step, released neurotransmitter molecules are deactivated either by reuptake or enzymatic degradation.
Dopamine is released by a neuron in the synapse where it can bind to dopamine receptors on neighboring neurons. Normally, dopamine is then recycled back into the transmitting neuron by a specialized protein called the dopamine transporter. Some drugs, like cocaine, instead of affecting the dopamine transporter protein, affect the neurotransmitter receptors directly. Dopamine agonists are drugs that basically bind to the dopamine receptors in place of dopamine and stimulate the dopamine receptors, even when someone has no dopamine neurons. An example where agonists are used pharmaceutically is for Parkinson's disease, since people with Parkinson's don't have any of the receptors for dopamine. So the agonists take a part in that and mimic dopamine to get the same effects as the neurotransmitter. In contrast to that, dopamine antagonists competitively bind to the neurotransmitter. They competitively bind to the dopamine receptors but don't stimulate the neurons. So they just suppress the effects of dopamine by not allowing it to react with the receptors. These dopamine antagonists lead to desensitization of the neurons, which is when the body doesn't feel the effects of dopamine, since the neurotransmitters that transfer the chemical signals are being blocked. So, to the nervous system, tries to make up for the less stimulation of the receptors by becoming more sensitive to dopamine. Such drugs are direct-acting long-term treatment with dopamine antagonists increases the number of dopamine receptors. In contrast, if dopamine antagonists repeatedly stimulate the dopamine receptors, the receptors become less sensitive to dopamine, which is called desensitization. Cocaine is a dopamine agonist, which remember is just a drug that competitively binds to something. There's an enzyme, MAO, monoamine oxidase, which breaks down dopamine and keeps dopamine levels fairly low to maintain homeostasis in our bodies. But if there is an extra amount of dopamine, the excess can be taken from the nerve ending back into the vesicle for storage. This recycling process happens from with the presence of a specialized protein, which is the dopamine transporter. When cocaine is present, it blocks the recycling process of the dopamine. As a result, more dopamine remains to stimulate the neurons, which causes prolonged feelings of pleasure and excitement. So this figure just shows how the cocaine blocks the dopamine transporter so that it can't be recycled back into the transporter and be stored in the vesicles. This results in a buildup of serotonin in the neural synapses. Methamphetamine plays the same role as cocaine in the brain with the effects of dopamine because it blocks the reuptake of neurotransmitters. So we'll be focusing on the effects of meth on the serotonin levels in the brain. Serotonin plays an important role in many behaviors, including sleep, appetite, memory, sexual behavior, and mood. Chemically, serotonin looks a lot like hallucinogenic drugs. Because of this fact, hallucinogen can bind onto serotonin receptors, mimicking the actual neurotransmitter. This results in unnatural stimulation in different areas of the brain. One way meth use affects the serotonin is by reducing the levels of chemical serotonin in the brain. The serotonin levels diminish in the body because meth blocks the synapse and prevents the release and production of serotonin from the synapse. Meth permanently affects the concentration of serotonin in the brain by reducing it because meth kills dopamine and serotonin neurotransmitter cells. This is irreversible because these nerve cells do not grow back with time. Due to this, the only way a person can mimic the feeling of having serotonin in their system is by using meth over and over again.
As mentioned before, the use of drugs suppresses the brain's production of neurotransmitters. We mainly focus on dopamine, but noradrenaline is also suppressed in the brain from the use of drugs. When a person stops taking the drug or the drug starts to wear off, the brain kicks back into action. Because of this rebound, a rush of adrenaline is released, and the release of adrenaline causes the symptoms of withdrawal. Many people think of withdrawal when they think of symptoms. There's two classes of symptoms, emotional and physical. Each category of drug produces its own set of symptoms, and the symptoms are different for every person. Here is a list of emotional and physical symptoms. Like I said before, different drugs cause different withdrawal symptoms. Alcohol, opiates, and tranquilizers produce more physical symptoms, while cocaine, ecstasy, and marijuana elicit more emotional symptoms. When a person is going through withdrawal, they will go through two different stages. The first stage is acute withdrawal, and this stage usually lasts for a few weeks, but the type of drug and the type of person varies the duration of the stage. During this stage, the person usually feels the effects of physical symptoms as the drug is detoxing from the body. Once acute withdrawal stage has passed, a person will enter the post-acute withdrawal, more commonly known as pause. This is the second stage of withdrawal, and most people go through a form of this stage. In this stage, the brain chemistry is trying to return to pre-drug chemical equilibrium. Because of the changes chemically in the brain, the person will usually experience more emotional and psychological symptoms. The symptoms experienced in the beginning will change extremely quickly, but as time goes on and the brain chemistry is closer to reaching normal levels again, the symptoms will subside and will change less. Some common symptoms of pause are mood swings, irritability, tiredness, and lack of enthusiasm. Pause usually lasts for about 2 years. This isn't a quick recovery, and the person has to be up for the challenge.